Alright guys, welcome to a tutorial. This is going to be your fifth tutorial, I believe, in your third unit, labeled the cell cycle. Okay, so we're covering the cell cycle today. And just so you guys know, I do have another tutorial up on Meiosis, and it's going to be extremely close to this one, so I'm not going to go into too much detail with this one. I'm just going to cover what I think you need for the mastery test. If you want more detail, make sure to head over to that Meiosis page, alright? Because that's going to cover a lot more than I'm going to cover here. In any case, let's go ahead and start with our tutorial. Let's start with, let's see, I think page 4. I don't know, I'm sorting through this. In any case, it is going to cover a lot of the same things. So let's take a look at just the fact that cells divide. And they divide into different cells via mitosis, and meiosis. There are two types of cells. One's called somatic. Soma means body. So these are your regular body cells. These are like skin cells, cheek cells, whatever else cells. And then you have sex cells. So sex cells divide a little bit differently than what you're going to be learning today. We're just going to talk about mitosis today. So mitosis is going to go through, or the whole cell cycle is going to go through 3 different parts. You've got interphase is part 1. And inter means between. So you've probably heard of intermittent or interstate. So the interstate is a road that goes between 2 states. So it's interphase is between phases. What this means is that this interphase, this is the growth period of a cell. Growth period. So it's like a cell is newly born or whatnot. This interphase is the time it takes for it to grow up big enough until it can divide on its own. It's kind of like thinking of a child. When parents have children, children don't just immediately have other children. They have to grow through that, 18, 20 years, whatever it is, until they're mature enough to have kids and all that jazz. So you have to go through that interphase. Interphase is kind of like childhood / teenagerhood. Teenagerism? I don't know. Whatever. It's that adolescence up until you're an adult. That is what interphase is. So it's that growth period. Okay? Following your interphase, you now have mitosis. So mitosis is where this is the actual relationship part. This is going to be where you're starting to grow. In this case, it's going to be the nucleus dividing. So during mitosis, your nucleus is actually going to divide. This is where you start to finally have all of everything splitting apart so that you can have more than one cell. So your growth period made sure that you have all the materials. Mitosis is where it actually starts to split apart. So now everything's packaged into 2 separate groups. And now you have cytokinesis. And with cytokinesis, all that is the cell finally splits apart. Okay? So cell splits apart. Cell divides. To draw this in a drawing, interphase is like you grow from a little cell to a bigger cell. Alright, so that's essentially what interphase is. Mitosis is essentially, you've got your big cell, but now you've got 2 nuclei. Alright, you've got 2 different parts in it. And you have 2 of everything, essentially, but I'm just going to draw the nucleus. Alright, so nucleus, another nucleus. Alright, so those are your 2 nuclei. And then over here with cytokinesis, by the way, this starts to furrow during mitosis. And then over here, you're going to finally have those 2 cells, each with their own nuclei. Alright, and now they're small again, so they have to grow big. But that is how mitosis works. Notice in mitosis, you're going to end up with smaller cells, so each one of these is half the size of a regular cell. Okay, it is half the size. That's why you have to go through interphase until you get to the regular size of a cell, so that you can split again. But that is the general idea with the cell cycle. Alright, hopefully that makes sense. We're going to go ahead. Oh, there's a drawing on here that's really, really nice. It talks about the different phases, but I think we're going to talk about them a little later. So on page 6, feel free to take a look at that diagram. It's quite nice. So let me go on. Let's see. Page 8 is going to talk about the phases. Okay, cool. We're going to start with phase 1 is G1. Alright, so the cell cycle has those 3 phases, but this is those 3 phases broken down even more. G1 is your growth phase. This is essentially what interphase is. This is like the main idea of interphase. There's one more part of interphase, but well, 2 more parts of interphase. But this is the main part. This is where your cell is growing. That's that drawing that I just did there. Now you're also going to have the S phase. S phase is a specific part of this growing. So yes, you're growing during G1, you're getting big, etc., etc. But S phase is the synthesis phase. Synthesis just means to make things, right? So what's going to happen is you're going to make doubles of everything. So you're just going to replicate all your stuff. All right. So the growth phase, you're just starting out, you're just getting some things. So this is getting some of the things you've lost. You know, so like let's say you just moved out of your parents' house. This is a phase where you just bought some plates, you bought some forks, you bought a couch, you bought a TV. You slowly bought things that you don't have anymore. Synthesis phase is where you're going to start doubling up. I think the example that I used in the meiosis video is a divorce. So like this growth phase, this is you moving out of your parents' house and buying your own crap. So you grow and you get your own crap. But then you get in a relationship and now you guys want to double your crap. So you synthesize. So you guys double your crap. Now your girlfriend brought in a TV with her or whatever. This kind of thing. So you make doubles of everything. Now, this is kind of a bad example. With meiosis, it works perfectly. With mitosis, divorce doesn't work as well because mitosis, you only need one cell. It just needs you. So I don't know why you would ever make double of everything. Yeah, I don't know. Human beings really don't do that when they're single. But in any case, pretend they do. S phase, you're going to make doubles of everything. Then you've got G2 phase. And G2 phase is where we're going to... keep in mind, you have half the DNA of a regular cell. Half the DNA of a regular cell. You have half of pretty much everything of a regular cell because you got to double it in the S phase. So during G1, you get half of everything. In G2, however, you're going to finally start to see a big difference. This is going to be the checkpoint. You're going to checkpoint to make sure, essentially make sure you have doubles of everything, but make sure you are ready to split. You are ready to divide. All right. I don't really have a very good analogy for this. Mitosis is a lot more straightforward than meiosis is. Meiosis, you can use analogies to understand it, but mitosis is really just kind of straightforward. So this G2 is a checkpoint to make sure you're ready to divide. If things are not ready at G2, you move back to G1 or S or something like that. But G2 is a checkpoint area, so that's probably the biggest point about G2. And then you're going to go through mitosis, which is, the actual splitting and all that jazz. It includes cytokinesis. Wow, sorry about that. Splitting of the cells. All right. So it's really just these 4 steps: G1, S, G2, and then mitosis. Mitosis is the splitting part, so that's the end of everything. So that makes perfect sense. In any case, that is your cell cycle, interphase, and mitosis. So we're going to move on to page 10. I totally forgot. I have markers and whatnot, so I should probably be writing those. I'll do that from now on. Let's see. So it talks about mitosis, and then now mitosis has its own phases, which they're going to describe here, and I'm going to describe a bit. Let's start with. I could do these in different colors. I'm going to start on a different sheet of paper. That way you guys have it, and I'm also going to write this landscape. I did this for the other section. It worked out pretty well, so let's go ahead and do the same for this one. You're going to start with prophase. Prophase is step 1. So meiosis is really just double this. Mitosis is just one of these. But anyway, prophase is just one step. So this is step 1. Alright, this is step 1, prophase. I don't know why I wrote this on the right. I should have written it on the left because, we read left or I don't know. Whatever. What happens is that you're going to form your spindles, and spindles are the things that hold the thread. So if you'll notice on the picture, it looks kind of like you'll notice that you have a cell, and then they always draw these like things, these like threads from all over the place. Whatever. That was really awful, but that's the idea. So those things are called spindle fibers. So in prophase, your spindles form. Spindles form. Alright, and then you'll get your centrioles. Centrioles are these little bundles that hold all the strings. So if the spindles are the strings, the centrioles are the things that hold the strings together. Alright, so the centrioles separate. They're just going to go to different ends. That way you can get your strings on these things, right? They have to be on different ends. If they were sitting right there in the middle, it wouldn't do any good. You wouldn't be able to split. So you've got to separate as far apart as possible, all right? And then you can see chromosomes, by the way. You can see chromosomes. Not literally. You have to like use a microscope on it. You can't just look at it. But still, idea remains. Now we're going to go into metaphase. So metaphase, the second phase here, is where now that you've separated everything and you've got all your chromosomes and whatnot, now you're going to get your chromosomes to all meet in the middle. Alright, so we still got our, whatever the heck, these spindle fibers, which mine are awful, but now your chromosomes are all going to meet in the middle. Alright? And you're going to have double of each. They're all going to meet, and they're all going to cross up. This is metaphase. You can tell because all your chromosomes are real close to each other. They're all generally touching. So chromosomes line up in the middle. Chromosomes in the middle. Alright? Middle. So metaphase, you can think of the middle phase. Has everything in the middle. Prophase, pro means before, so this is the before part where everything separates. Metaphase, middle, alright. And then now let's talk about anaphase. I'm going to use green for this, I guess. Actually, here. Orange. So here is anaphase. Alright, anaphase is going to be where everything separates. So they were in the middle, now all of my new chromosomes are going to separate apart. And keep in mind, they are different now. They're not all the same. Alright, so they're still on spindles. I'm just not going to draw them for sake of whatever. So my chromosomes are going to start separating. They're going to go outwards away from each other. Okay, so chromosomes separate. Alright, that's the main thing in anaphase. And, both sides have their set of chromosomes. And now we're going to move on to our final phase, telophase, which is also the cytokinesis phase, but they're slightly different, but the general idea is that it's the last phase. And what's going to happen is that you start to see cleavage furrow. And what that means is that you start to see the cell start to pinch on itself. So now that they've got their chromosomes, they start to do this kind of thing. Alright, so they're going to start to split apart because you've got, this one's got its chromosomes, it's happy, this one's got its chromosomes, it's happy. So they're going to start to separate. So this is prepare for cytokinesis. And the last step, I guess, is technically cytokinesis, but it's not considered part of mitosis exactly. So they prepare for cytokinesis. Okay, so these are the 4 steps in a very general way. Like I said, there is another tutorial with meiosis where I go in detail with these. But for now, prophase 1, metaphase, anaphase, telophase. It's just the 4 steps in order. You got to know what happens in each of them. Okay, so let me just slowly get them all on screen. That way that you guys have them. So there's prophase and metaphase. Feel free to pause here. There's anaphase, telophase. Feel free to pause here. Alright, so those are the steps. Let me go ahead and see what's on the next page while you guys are copying. Anyway, while this loads, this is all fairly basic information. Like, you guys already know that cells divide and split apart and all that jazz. You just have to know what they're all called now. So page 12 has an activity for mitosis. Page 13 is going to talk about the actual cytokinesis thing. I told you guys, cytokinesis is just when the cell splits. Keep in mind that your regular cells sit at this large phase. So after cytokinesis, remember that they're half as big. So you have to go through this G phase again to get bigger. So during G phase, you have half the things you should have because you're starting with what you ended up with in some other process. So G1 specifically, you have half the stuff as you did earlier. Alright. So, move on, page 14, another activity talking about cytokinesis. And then 15 is 15 some kind of table. Oh, talks about plant versus animal cells. So just remember, there are certain things in plants that animal cells don't have. The one thing I want you to note is that plants have that cell plate or that cell wall. So rather than a metaphase plate that we have, plants form a cell plate. That's the one major difference between plants and animals. There's really no other big difference. Like, the centrioles work a little bit differently than the animal cells and, whatever. But they've got that cell wall, and because of that hard cell wall, because plants are a little bit crunchier than humans are and all that jazz, they do have a cell plate that forms as opposed to our median plate. Okay, so just keep that in mind. Really not that big a deal. It's one thing. plants have a cell plate, and that's all I really need you to remember. There are like minor differences. We're not going to go over all that. So let me go ahead. Page 16 is just another activity. 17. Okay. Cool. So yeah, let's just go ahead and review because all it is doing, it's going to do a summary and all that jazz. And it's loading weird. I thought I heard a weird ringing. Anyway, so G1 in the interphase. So you've got these three phases: interphase, mitosis, and cytokinesis. All right. And during G1, you're going to have half the DNA of a regular cell until it grows. And once it grows, you're going to synthesize and you're going to double everything up. Now you've got the regular stuff of a cell. Cells have like double and everything. So you're at the regular point of a regular cell once you hit S phase. Then now here during G2, you're going to make sure you're ready for everything. Mitosis is going to split. All right. In plant cells, you're going to have a cell plate. So rather than metaphase, this metaphase plate, you're actually going to have like a literal hard plate here. That's going to be the cell plate for a plant. All right. But generally, you can go over, or you can use these these 4 phases to talk about mitosis in this way. So yeah, just keep all those 4 in mind, and keep in mind what order they go through. But yeah, I think that's pretty much it for mitosis. Did I read another page? No, that's it. So mitosis is pretty short. Like I said, if you need any more, you can always go to the meiosis meiosis tutorial in Unit 4. It'll all eventually be in this playlist. So let me know if the master test give you guys trouble, and I'll see you guys next video. Ciao.